UC Berkeley with Jim McGuire, but this is a project I worked on primarily during my master's at Villanova University with Todd Jackman and Tony Gamble. So we essentially built these, put together this new set of loci in order to deal with. We basically wanted the best possible individual loci for phylogenetics that we could get, and because essentially marker selection has emerged as a major issue in phylogenomics. You know, as I'm sure many of you know, you have to balance all these different trade-offs when you start deciding on different markers. Should you choose coding or non-coding markers, conserved or highly variable ones, long or short ones, SIPs by themselves or full sequence alignments, and then also different types of markers like that are potentially homoplazy-free, like transposable elements or microRNAs. So despite kind of going into the genomic era now, we still are dealing with marker selection because aligned sets of orthologous loci are required for phylogenomics. So even with full genomes, you have to subset those genomes in order to build your species trees from that. So we wanted to design the best set of loci that we could for these phylogenomic analyses. So as many of you probably know about this, a major comparison between different data types was this Jarvis et al. Paper from 2014 in Science, where they looked at 48 different bird genomes and compared different types of data, and we got vastly different trees depending on what data they used. This was later kind of looked at in more detail by Reddy et al. In 2017, and they found that model violations actually best explain the discordance between these different data types. So you have non-coding data showing one kind of general tree and coding data showing another type of tree. But it was unclear whether non-coding or coding data was a greater violator of the models. And so it's still up in the air, you know, what type of data are both, or potentially both, are violating models. So when gene trees don't match the species tree, that's when we have the most difficulty estimating it. And these are the cases where we want the best loci possible. So gene tree discordance can be caused by a number of things. Systematic error, like I just mentioned in the previous example, where your models may not fit your data very well. You can have incomplete lineage sorting and regression, but these are probably the case where we have the best analyses to try to deal with them. Like coalescent species tree analyses are pretty good at dealing with ILS. We have new analyses being invented every year for dealing with introgression as well. But a third issue is gene tree estimation error, and this is really where we focused our attention, trying to have really high quality individual gene trees. I'm going to go into this a little more detail here. So gene tree estimation error is when phylogenetic information is limited, the gene tree may not show the true genealogy. So this is most likely to occur with short loci and those with slow rates, because you just simply don't have enough information there. For example, some UCEs, especially short ones, and short exons might not have enough information in them. Many studies use exon capture, but they end up with very, very short exons that might not give very good gene trees by themselves. So there's been some solutions to deal with these problems, like statistical binning. You can join together different loci that show similar histories and build better gene trees from those concatenated sets, and then build species trees from those. Or you can filter loci based on the quality of the gene tree resolution or phylogenetic signal to basically just pick out the best loci that you've sequenced for your question of interest. But these are we kind of consider these stopgap solutions, when really, why don't we just sequence the best loci we can from the get-go and focus our attention on those. So that's why we developed rapidly evolving long exon capture, which we call RELEC. The idea is essentially to find single exons that fit these criteria. They're at least 1,500 bases in length. We wanted them to be long enough to hold tons of phylogenetic signal in them. We wanted them to be more rapidly evolving than RAG1, which is a commonly used marker that in our experience provides pretty good gene trees in general. It has enough variation to do that. We wanted them to be, all these markers to be present in all amnio groups so that we could combine different analyses or different studies into meta-analyses. And we also wanted to avoid recent paralogs. So any gene duplication events that might kind of muddy the phylogenetic history, we just wanted to avoid all of that. So basically the idea is of RELEC is to maximize genetic variation while maintaining presence orthology across a broad range of evolutionary scales. So there are many sequence capture data sets that have been developed in vertebrates, and which I'm going to be focusing on in this talk. The most common methods that target orthologous loci across all the taxa in which they're utilized are ultra-conserved elements and anchored hybrid enrichment. So I'll go into these in more detail. There are many other methods that have been used, but I'm just not going to, for the sake of time, not going to really talk about them too much in this talk. So UCEs and anchored hybrid enrichment. You just heard about UCEs in the last talk. I think there's some more ultra-conserved elements talks coming up. They've been very successful. Both of these have been very successful at solving some nodes, but you know, there are still many cases in which when they're used, they can't actually fully resolve a species tree, or there's certain nodes that are still ambiguous. So even though these have been pretty good, we wanted the best loci as possible, which is why we developed Relic. So UCEs have about, I'm just talking about amniotes and vertebrates. In these cases, these have been developed for other organisms as well. But UCEs have about 5,000 loci captured using these conserved central core regions with 100 to 200 base pair probes. They're almost all non-coding. There's a few that are in exons. And they work across all amniotes. In general, UCEs target the flanking regions around these conserved probe regions. Anchored hybrid enrichment has about 400 to 500 loci, depending on the version. They target much longer regions. They are primarily now in version 2 almost all on exons, though they do target flanking regions and introns as well. And they work across a broader taxonomic scale in all vertebrates, so that includes fish, fish and amphibians. So the benefits of Relic that are essentially, the major one is that we have abundant phylogenetic signal, and this decreases the chance of gene tree estimation error. It makes each individual gene tree very, very high quality. The rest of these benefits here are benefits of using exons in general. You can model evolution potentially more accurately. You can, many of the protein functions are known, so this could allow for studies of functional differences and amino acid replacements. Alignment of protein coding regions is straightforward using translation alignments. Indels can also be valuable phylogenetic characters and are more easily modeled in exons in these 3 base pair intervals. So to compare Relic with UCEs and anchored hybrid enrichment, we looked at 17 different squamate genomes, 2 of which we sequenced, and we also sequenced 1 transcriptome to confirm all the Relic genes are expressed as mRNA. So in total, we had 10 snakes, 4 geckos, 2 iguanians, and 1 anguid. This kind of spans a broad range of taxonomic scales, from more recently diverged snakes to the more deeply diverged all these snakes and lizards. So just to briefly go over the methods, I designed this this program, orthologic assembly and concatenation, to basically extract all the loci from the genomes. We were having some issues with other methods, so I'll talk about this, how this works in a second. We built alignments with MAFT. We confirmed the translation frame for each alignment manually in Genius for all the relic loci. We built gene trees in Genius, and then species tree primarily in ASCII 3, and we also built a full Bayesian coalescent tree in Starbase 2 for the relic loci. UCE data set was extracted with Phylogeny, which worked really well for the UCEs, but didn't work so well for these divergent, these rapidly evolving relic genes. So the oak pipeline we developed because essentially when we were trying to blast these genes out of the genome, we were getting really segmented blast hits. So if you imagine this is a genomic contig, and this is your target exon on that contig, when you query that exon, if there are rapidly evolving segments within it, you'll get these segmented hits. And we wanted to be able to extract the whole exon from that. So essentially what we did, I built this little program to basically pull the min and max of those blast hits from neighboring areas on the same contig. We pulled that out, we added a little bit of flanking sequence, aligned that back to the query sequence, and then trimmed off the ends. So that gives us the complete ortholog sequence. So if you're interested in this program, you could use it to pull relic out of new genomes or other sequences as well. There's a translated blast option and a regular blast option. So these scripts are available on my GitHub page. Oops. So the rest of the talk I'm going to be comparing relic versus anchored hybrid enrichment versus UCEs. So we pulled these out of each of those, all those genomes. Here you have the number of markers for each data set and the total number of aligned bases. That's what you can focus on here. We wanted complete data matrices with all the taxa, just to avoid any issues with missing data. So we got 179 relic loci. They're very long, so they total a large number of bases. 320 out of about 400 anchored loci, and then about 1,500 UCEs. There's a lot of missing data in some of these genomes, and that accounts for why we didn't get kind of the full number in there, because we wanted to really just have a complete data matrix. But these data sets should be pretty comparable, and so we compared them using different methods for different measures of phylogenetic formaveness and gene tree quality. So first here you can see kind of a general comparison of the length of each of the loci in each data set. Relic is in green, anchored in orange, and UCEs in purple. You can see relic genes go from about 1,500 bases all the way up to almost 13,000. Some of them are really, really long. And then here we have on the left the proportion of parsimony for additive sites within each of those loci. And parsimony informative sites are the sites actually used in, actually useful for phylogenetic analyses. They have the information, the phylogenetic signal you need. And you can see per unit length, relic loci have much, much more phylogenetic signal. Here's RAG1 for a reference. The most anchored and UCE loci have fewer than RAG1 per unit length. Here is the tetrapod phylogeny, and showing the number of relic loci within each of these model species genomes. We went out to tetrapods just to kind of look at how much drop-off we get, but this is not really the focus of relic. We really are trying to focus within amniotes. So on the top here, you can see, there's a few exceptions where there's been a deletion within one of these genomes, but essentially they are present across all these genomes. We allowed a few cases of gene duplications for some loci that we really wanted to include because they're very informative. and that accounts for some of the differences in numbers here. I was kind of surprised that about the annotation. Here in red is the percent of the genes that are correctly annotated on Ensemble. And actually, only in humans do we have all the correct annotations, which you would expect. But in all these other lineages, we had a lot of errors. As I went through it, there were exons that were split up incorrectly into multiple parts, or the annotation was just fully missing. This is probably just because these genes are so rapidly evolving compared to, other genes, that these annotation pipelines might not work so well for them. Similarly to like how blast searches don't perform perfectly. Yeah, so it wasn't quite as easy as you might expect to pull all these genes together, but we managed to do it. So here I'm comparing the utility of each of these data sets across the squamate species tree here. So this squamate species tree is pretty well resolved. There's not that many nodes that are really contentious. So it's a good tree to look at. And all the data sets resolve this tree pretty well. This is the combined species tree for all three. We had one node different in the anchored tree, but it was at a node with low support. But essentially, they're all resolving the same tree. And what I want to draw your attention to on this graph are these numbers on the left of the nodes. This shows the proportion of individual gene trees that match the species tree for that node. And you can see here that for nodes deeper in the tree, Relic outperforms, Relic was on the left, anchored in the middle, and UCEs on the right. Relic will outperform both of those other methods for individual nodes. Some nodes are very well resolved for everything, but then as you go up in the tree, you see this pattern very, very strikingly at these more recent nodes. So we think so far, Relic gene trees look really, really good, really high performing at these individual nodes. We also looked at the phylogenetic informativeness of Relic compared to anchored and UCEs. This is the power of the gene to resolve a node at any given point in time. So using that squamate tree, you can estimate these curves. And we can see Relic far outperforms anchored and UCEs on this metric. Tons of phylogenetic signal. And this is likely basically due to this very strong correlation between phylogenetic informativeness and alignment length. So as you get longer alignments, you get more phylogenetic signal. It makes sense. We also notice here that the time of maximum phylogenetic informativeness is much more recent for Relic compared to anchored and UCEs. And this is useful for people doing studies on more recently diverging taxa. You want your loci to be tuned to the your group of interest. So here's another metric. I'll just go through this pretty quick. So this is the RF distance, or R. Robinson's fold distance of the gene trees from the species tree. And you can see, as you compare the different data sets, Relic is skewed to the left compared to these other data sets, showing that the whole tree is more similar to the species tree than in Relic versus the other methods. And this is especially apparent in the more recently diverging snake lineage, the colubrid snakes, where you have almost 90% of Relic loci perfectly matching the species tree, whereas only about half of anchored and less than 20% of UCEs perfectly match that subtree. I'm just going to skip over this for the sake of time, but you can see here that the Relic genes also have a higher mean bootstrap score compared to anchored and UCEs, which is another indicator of gene tree quality. So a few potential concerns with Relic that have come up are substitution, saturation, recombination, and selection. I'm going to show you some evidence here that these shouldn't be major concerns. So with rapidly evolving genes, substitution, saturation, you could imagine might mask the phylogenetic signal within those genes if you have multiple mutations at the same sites. This is mitochondrial ND2. You can see this strong signature of saturation as you get deeper in time. These are split up by the codon positions shown here. In relic loci, you see very linear divergences through time, with some loci getting some saturation deep into the tree, but most of them show linear plots all the way to the 200 million years in the study. Other loci actually show similar linear plots for all the codon positions, indicating really strong or really relaxed selection on these loci. Here's the BRCA1, which is also a breast cancer gene in humans, and it shows very relaxed selection compared to other genes. So relic genes are long, so there could be some recombination breakpoints within them. This would be a problem for some analyses, but we don't really expect it to be a major problem because it seems like these recombination causing incomplete lineage sorting on either side of that breakpoint seems to be rare. We also see that they, I also looked at the map distance between the loci on the anomalous chromosomes, and you can see this plot here showing where they're located, as well as the GC content for each of these genes. And we find that relic loci are very rarely within 50,000 bases of each other. There's only 5 loci that get that close, whereas UCEs can get within, there's actually 20% of the UCEs were within 10,000 bases of each other, and 50% were within 50,000. So this was kind of a surprising result for me. I think potentially there could be some linkage between loci when looking at UCEs, and I think this is something people should look into in the future. And if anyone's interested in collaborating on this kind of topic, I'd be definitely interested in looking into this more. So I'm almost finished here. So looking at selection, relic loci show so relaxed purifying selection in general, with mean D and DS ratios around 0.5, compared to a means of 0.15 to 0.3 across coding sequences in other organisms. So this makes sense that they have some, their coding regions, they should have some negative selection, but it's overall relaxed compared to most. I just want to emphasize here that UCEs are definitely undergoing strong purifying selection, up to 3 times higher than protein coding regions. So relic coding regions shouldn't be the primary focus of arguments about selection in phylogenomics. But an advantage of using coding regions is that you can model nucleotide evolution and the strength of selection really well with using these D and DS numbers. So this is really a benefit in general to using these markers. Just want to mention really quick that we were able, because we have fewer loci, we're actually able to utilize these full Bayesian coalescent species tree methods, which are really preferable. They simultaneously model the gene tree and the species tree, and they can get better estimates of the species tree. So the main limiter on these analyses is the number of loci. And by having fewer loci that are higher quality, you can actually run these analyses. So if you're interested in using Relic, here's some information for you. The targets are about 700,000 bases long. You can use about 11,120 base pair probes from MyBaits. We've already done this successfully in geckos. We get high coverage across deep divergences. They don't seem to be a problem capturing them up to 55 million years. So we're testing these in other more recently diverged groups to kind of show if Relic works well at the tips of the trees. Where target loci are currently available for all amnio groups on my GitHub page, and we're working potentially on building new data sets in other groups as well, for example, arthropods and others. So to finish up here, Relic loci hold abundant phylogenetic signal and may help to resolve some difficult nodes in the tree of life. They can be used alone or can complement other sequence capture data sets. For example, UCEs and Relic would be a really powerful combination with lots of phylogenetic signal. So if you're doing phylogenomics, consider also capturing the relic loci. I'd like to acknowledge Bauer and Jackman Labs and McGuire and Bowie Labs at UC Berkeley for help on the manuscript and all these contributors of these photos. That I could take any questions.
So today I'm going to present to you part of the work I've been doing in the California Academy of Sciences. Like Athena said, I just arrived at the Cal Academy a couple months ago before the quarantine. But I've been working this project in collaboration with Cal Academy for over 2 years now. So this is some of our results. And I'll be talking today about diversity, species diversity, and species diversity patterns. And a lot of the time, species diversity is inferred by species accounts, by the number of species, or species density, how many species you have over some area in space. And we have several studies that have been showing us how the species are distributed in the world and how much diversity we have. Here you can see a map showing the species density for ants all over the world, with the warmer colors showing the higher areas of diversity, the areas that have the larger number of species. And I want to point out here that the tropics usually are the areas that have the most, the higher number of species of ants in the world. And among those areas, there's Madagascar, where, as you can see, has a very, very high number of species all across the island. And it's a very interesting system with a lot of very striking habitats. But biodiversity is not only about the number of species. If you can see here, if you have plot A and plot B with 6 species each, we could say that they have the same diversity. But if you look at the photos, you see that they are very different from each other. And this accounts for diversity too, because diversity shouldn't be only about species numbers, but should be also about the relationships and the higher clades. So if we talk about species, we're only considering one level of clades, but there's many other levels that we can consider, like genus or families or orders. And all this has an influence on diversity. So we can account for phylogenetic diversity in different areas if we want to use evolutionary data to also infer diversity. So I want to tell you about some terms that I'll be using in this presentation today, and talk to you about what I mean about phylogenetic diversity. So if you have plot A, here we have 6 species, but we can say that we have a low phylogenetic diversity, because if you see here, they're all the species are closely related. So we can say that we have less evolutionary history in this plot A, which means that we have more shared branches among the species and more recent lineages. And I'll be calling this phylogenetic clustering throughout the presentation. But if we look at plot B, we also have the 6 species, but they're not the same species. And this plot B actually has a high phylogenetic diversity, because as you can see here, you have the species distributed all over the phylogeny for the ants here that I showed you, which means that we have more evolutionary history in this plot. We have fewer shared branches. They're more distantly related and more ancient lineages. And this is what we call phylogenetic overdispersion. And in the same way, endemism is also not about just species. It's not only about which species are unique to an area, but we can also account for phylogenetic endemism when we include phylogenetic data into those inferences. The phylogenetic endemism is an index of localized evolutionary history. So similar to species endemism, phylogenetic endemism show us the areas that have unique evolutionary history contained in them. And using that type of metric, we can calculate ranges for each branch and not each species, which would allow us to identify centers of endemism in different areas. Like this work here by Wilme and collaborators, it was published in Science, that show us centers of endemisms for ants, for lemurs in Madagascar that are very interesting and show us which areas are more important for each species. So with that in mind, this project, MAMI, which is Madagascar Microendemism, has been conducted, funded by NSF, is a project in collaboration with Dr. Bonnie Blamer from previously the North Carolina State University, and now at the National Museum in Berlin. Dr. Brian Fisher, the California Academy of Sciences, I'm sure you guys know him, and Carol Loss, which was a postdoc in the California Academy of Sciences, now working on the National Institute for the Atlantic Forest in Brazil. So the goals of this project are to describe patterns of species richness, phylogenetic diversity, and endemism, and phylogenetic endemism also for the ants of Madagascar, and to identify centers of diversity and endemism important for arthropod conservation in Madagascar. So we can use ants as surrogates for centers that are important for arthropod conservation in the island. And why Madagascar? Like I told you, it's an area that has a very high diversity of ants. It also contains very high habitat diversity, as we can find dry forests, subhumid forests, rainforests, and even unique biomes like the spiny forests that occur in the south of Madagascar. And this high diversity is contained in a small scale. It's just an island that it's isolated from the mainland for a long time. So there has a long history of isolation with very high levels of diversity and of endemism. So at least 50% of the species of ants in Madagascar are endemic to the island. So this is a very high number. So that makes Madagascar a very interesting system for us to study. And just showing the spiny forests here. And also, we know very well the ant fauna in Madagascar. The ant fauna there is very well known and well documented. Dr. Brian Fisher and his team has been collecting in Madagascar for the past 25 years broadly. And this is a huge database in AntWeb that most of you probably are familiar with. And ants have been collected all over the island, as you can see with those white dots here. Those are the points where we have ant collections for the island. So it's a very well-sampled area. And I generated data for 1,268 species of ants. These are all the ants of Madagascar that we could get a hold on. There's some that are missing because they're only known from holotypes that are not deposited in Cal Academy. I already generated actually 1429 terminals. We have a few species that we did it again, so it's a very large data set. And of those, 627 are endemic species that only occur in Madagascar. And we have more than 140,000 collection records for those species in Madagascar. And with those ants, I generated a phylogenomic data set. So we used a non-destructive approach, soaking the ants in solution to extract the DNA from them without destroying them, so we can keep the vouchers for the specimens that have been extracted, so we can confirm species identities later. This was generated for all the 1429 terminals that I told you. I have an average of 2,200 UC loci for each species, which is a very good number, especially considering that most of those ants are 20 or 15 years old, and using a non-destructive approach. So those are really good numbers for our data set. And the final goal is to generate an ant community phylogeny, so a phylogeny for all the ants in the island. we've been making some progress in this, and the preliminary analysis for all lands in Madagascar are showing very good support and very well resolved topologies. So I'm very happy with that. The challenge now is to construct a alignment for this entire data set, which has been proving very tricky. So if anyone has any tips for me, I would appreciate it. And I'm combining this phylogenetic data set, this phylogenomic data set, with models for species distribution on the island. So from all those collection records we have, we used almost 70,000 of them. We curated them for the records that are more specific and that we could trust more from 2,700 localities in Madagascar, those that I showed you before. And we combine this with environmental data to model species based on a climatic niche data set and generate those expected distributions for the ants, for each species of ants in Madagascar. And we did, Carol Laws actually did that using Maxent in R. And what I'm going to share with you today are our first results on Chromatogaster diversity. So I picked a genus to show you guys how those patterns are emerging. For Chromatogaster, I had 34 species of Chromatogaster, all species that occur in Madagascar except one. And I generated a phylogeny, maximum likelihood phylogeny with IQ3, and use this and the alignment as an input in MCMC3 with 3 different calibrations, secondary calibrations, to then generate a dated chronogram for Chromatogaster in the island. And this topology recovered the same 5 groups that a much larger work from Bonnie Blamer showing the same species groups that she showed in her work for Chromatogaster. We can recover them here for Madagascar. And using this dated phylogeny and the expected distribution, the model data for Chromatogaster, we were able to identify some patterns of richness, species richness, and phylogenetic diversity for Chromatogaster. So if you can look here, the left map in the left, to generate those analyses, I used the R biodiverse pipeline. This is a very interesting method. And you can see that the taxon richness for Chromatogaster in Madagascar has a very striking pattern. So the warmer colors are the areas that have the higher number of species. And here in the northwestern part of the island, we can see a high spot of diversity, and in the center of Madagascar too, while in the South Madagascar, you have a lower species diversity. But when we look at the phylogenetic diversity of those with the same data, you can see that there's actually some dissimilarities between the data. So here in this northwestern part, you can see that it's an area that has a lot of species, but also a lot of phylogenetic diversity. While in the central area of Madagascar, we have a lot of taxon richness, but not a lot of phylogenetic diversity. And this is a very interesting pattern to see. I want to point out the phylogenetic diversity is highly correlated to taxon richness. So this it's a very similar pattern. But if you think about it, this makes those differences even more striking. And if we randomize this data and look for the significance of the phylogenetic diversity in different areas, we can see which areas have significantly high phylogenetic diversity, which are the areas here in red and orange. So the areas that have high taxon richness and high phylogenetic diversity here in the north of Madagascar are recovered as areas of significantly high phylogenetic diversity. So like I told you in the beginning, we're calling this phylogenetic overdispersion. This means that in those areas here in red and orange, we have fewer shared branches and more evolutionary history in those areas. While in the center here, where we had high taxon richness, but low not so high phylogenetic diversity, we can actually see in blue and light blue that those areas have a significantly low phylogenetic diversity. So those areas show phylogenetic clustering, which means that we have more shared branches and less evolutionary history in those areas. If we look at the patterns of endemism for Chromatogaster, we can see the weight endemism, which is just the number of species that are endemic to those areas, and the phylogenetic endemism, which is this index of phylogenetic uniqueness for the branches. And we can see that those two maps are very similar. There's not a lot of endemism for Chromatogaster in most of Madagascar, except for those areas here in the north, that in different shades of purple, and they are very similar. So the areas that have species endemism and phylogenetic endemisms are very similar. But using those data, we can also calculate the areas that are significantly high on phylogenetic endemism and find centers of endemism for Chromatogaster in Madagascar. So I'm going to show you this graph this map a little closer. You can see here in the areas in red that we have what we call neo-endemism, which is a high concentration of short branches, a significantly high concentration of short branches, which could mean that those areas harbor species that are recently risen, either due to reproductive isolation or to hybridization. Those areas in blue are recovered as centers of paleoendemism, so areas that have significantly high long branches. So those are areas where we have species that were formerly widespread, but now are restricted to those areas. And the areas in light green show a mixed, what we call mixed endemism. So it's a mix of long and short branches, significantly high number of both short and long branches. And in dark green, we can see the areas that we call super endemism, which are areas that have significantly high level of long and short branches, so a mix of those two. And as you can see, the same area that shows very high phylogenetic diversity that are over dispersed are also areas that have both mixed super and paleoendemism. So areas that have a lot of a high diversity of branches that are longer than the other areas. So if we look at those centers of endemism for Chromatogaster in Madagascar, we can see how they are related to each other. So this is a cluster dendrogram, not a phylogeny, that shows for each tip of this dendrogram, we have a little square here in the map, and different colors in the map relate to the colors in the dendrogram. And here the dendrogram shows which areas are closely related to each other. So those areas here in the north of the island and in the east of the island, as well as those in the extreme west here, are more closely related, have more similar levels of phylogenetic diversity than those areas here in the center and in the south in orange and purple. And those centers of endemism are very similar to those ones that are found for lemurs, reptiles, and amphibians, which shows that those areas have something in them that are harboring those different, those endemic species for very different groups. And here in this hashed black areas are the protected areas of natural vegetation in Madagascar. So we can see that there is some overlap with our areas of high endemism for Chromatogaster, but not a lot. So there's still a lot that would need to be protected to protect those unique species and those unique lineages of Chromatogaster in Madagascar. And just to point out, I want to show you that there is also very little overlap of those centers of endemism with the primary vegetation cover that is still remaining on the island. We know that there's a lot of there's a lot of deforestation in Madagascar. There's very little primary vegetation left, and those centers are not in those areas of primary vegetation. So this means that we probably already lost a lot of this diversity now, because if you remember, those are the estimated ranges of distribution of those species. So that shows us that we are losing a lot of unique lineages in Madagascar, which points out how important it is for us to propose conservation measures for the island. So the take-home messages that I want to leave is that for the montane forests in the northern, northwestern, and south central Madagascar appears as significant centers of phylodiversity and endemism for Chromatogaster. We're very curious to see how those patterns are for all lengths in Madagascar. There's a partial overlap with centers of endemism for lemurs, reptiles, and amphibians, which we are also curious to see if that holds for all lengths in the island. And much of the estimated endemism is included in protected areas, but there's important gaps remaining that we need to consider in future conservation measures for the island. And like I mentioned before, our next steps are to define those areas of ant diversity and endemism on an island wild scale for all ants. I have the phylogenomic data for this already, but I don't know. I'm still working on generating the phylogenetic diversity and phylogenetic endemism. So I hope I'll have new results for you soon. We want to be able to do recommendations for arthropod conservation using the whole ant data set in the future. And I hope this is an interesting result of our work. And we also will be able to define areas that are undersampled in Madagascar, not only in the number of species, but also in the phylogenetic diversity and phylogenetic endemism that they contain. So those are our main goals of our MAMI project that we hope to achieve by the end of this project. And with that, I'd like to thank both my labs, the Blamer Lab and the Fisher Lab, and all the members of it, the Madagascar Biodiversity Center, which without who we would not be able to do this work, and all the funding agencies and institutions that harbor me. Thank you very much.
Thank you so much, Gabi. You have some questions. So Helly asks, how do you know a species was formerly widespread?
So this is an inference that we make based on the fact that we have a high number of very long branches in this area. So if we have very long branches, this means that the species has a long evolutionary history. So the inference that we can make when we have a significantly high level of long branches on those areas is that those are probably species that were widespread before and now are contained in those small areas.
Nice. And Ali has a question.
I do. Hello, Gabi. Great talk.
Hi, Ali.
I was just wondering, it's super interesting that you are considering the phylogenetic diversity, and I was wondering, in some ecosystems, marine mostly, as far as I know, usually when you have high phylogenetic diversity, you also have systems that have functional redundancy, and when they face a disturbance, they usually are really good in coming back to the initial point. So I was wondering if you are considering to evaluate that later in your project.
Yeah, that's a very interesting question that we want to be able to answer. this, like I said, our preliminary study that we're working on. So when we have all ants and we have a much more fine reading of those areas, we'll be able to tell if that's true. And that's definitely an interesting result that I hope I'll be able to answer with, especially with the whole data set.
And I do have another one, if you let me, Athena.
Yeah, go for it.
Gabi, so what variable do you think is actually running that diversity? Because you have a bunch of them, and I don't know if I missed it, but do you have any hypothesis about what's driving that high phylogenetic diversity in those spots?
No, not so far, to be honest. Those areas that have a very high phylogenetic diversity are also areas that have very high endemism, so have very unique species. So I think those are factors that are interfering there. And it's also montane areas, so there are areas that are more preserved because they are more difficult to reach, so they end up not being taken away. So those could be factors that are interfering in that.
Matt has a question. Matt, do you want to ask your own question?
I'll turn my mic on.
You can just yell.
How does the phylodiversity compare to the continent? And are there any really good dispersers?
I am not familiar with any studies that show the phylodiversity for ants in Africa. The Madagascar is very isolated from Africa for a long time, so the biggest similarities from the phylogenetic diversity, I think, would be to areas in Asia, maybe. But this is an interesting question that it would definitely be interesting to notice. We do have some species from Africa in my main phylogeny that were included for some groups, but not for all. So in broader studies that study different genus or the family, you would be able to see if there is a lot of dispersion events from Africa to Madagascar or from Madagascar to Africa. But since our data sets only has the ants from Madagascar, it's hard to tell.
Great, thank you. And thanks to all the speakers. Really great talk today. I think it's like a really good sampler of what type of questions you can ask with UCE, even within just one group. So please come back next week. We have more target capture talks. Very sadly, no more ants, but we have 6 great talks on penguins, spiders, beetle, rock daisy, and many more. Thank you, everybody, for being here.